On breeder wish list. Why to bother about breeder wish list? Because they are probably the first and main client for any phenotyping activity what we do, particularly working in a crop improvement program. So breeder wish list is a kind of take-home message, a conclusion slide which I deliberately put here. This strategy I learned very recently. If I'm placed at the this late hour of the day, so better to put at the beginning. So it's not non-exhaustive, of course. So the minimum requirement, and most of the breeder, whether it is molecular breeder or conventional breeder, they expect these things from us, that minimize experimental error and improve heritability. If it is abiotic stress trial like drought, heat, so heritability is 4.4 or more, and if it is optimal condition, so it should be 0.7 or more. Of course, in our CIMIT managed trial, we hit 0.6, 0.7 in case of even abiotic stress trial, but remember we deal with a lot of partners in developing world, so we don't expect very high heritability, but this is a minimum cutoff. Precisely estimated account for genotype by environment in selection, because so that's where we have to be very careful in terms of phenotyping site. Tool for improving precision and efficiency of data collection on key agronomic traits or phenotyping traits. So here we are talking about precision and efficiency, which means we are talking about high throughput precision phenotyping, but probably precision comes before throughput in this case. Traits are heritable genetically, not just phenotypically, correlated with yield and eventually adding in selection gain. Any trait which is not adding in selection genetic gain, I think they are not going to pick up and it's not going to help anyway. And in this era of genomics, molecular breeding, we have additional things to do is like dissect complex trait into components. It is useful for genomic region discovery. Let's remind the importance of precision phenotyping, whether it is conventional breeding or molecular breeding. It is actually key of the success for any genetic improvement program, and it is placed in the center. If it is conventional breeding program, what breeder do? They look at the diverse germ plots, phenotype them, select and feed to the breeding program for developing improved product. If it is molecular breeding, they just go there, genotype, and again, without phenotyping, there is no headway, and it goes and identify association analysis, for example, GWAS and promising alleles are fed to the breeding program. So it is in the center of any crop improvement program, irrespective of what approach is followed. We are in era of genomics. It's evolving very fast, starting from this era of RFLP, SSR. Now we are talking about full genome sequencing, and it's like a rat race, epigenome, methylome, and whatnot. Looking at this progress within just MACE program in CIMIT, in 2009, there was probably 500,000 data points, and by now, it's more than 7 billion data points. So it's really very fast. But if you compare this, and this is all has to be supported by high quality phenotyping data. So it sounds like it's a huge mismatch between genotyping and the capacity of phenotyping we have. So these statements like a significant mismatch, we are able to study gene and manipulate them, but the study of the process at level of whole plant has not been able to keep pace with these advances. So this is a huge challenge, and that growing disparity between, in terms of cost and speed, with genomics and high quality phenotyping is the major impediment in functional genomics. So these are some facts and which we have to deal with. As I said, it's an era of genomics, so it sounds like we have a lot of information at genomics level, whole genomes is sequenced, so we have kind of known genotype, and we expect this genotype to be translated into known phenotype. But in practice, it never happened. We know the genotype, we know the gene, and we know the whole genome sequence, But in between, there is something called environment, which includes prevailing weather condition, of course, soil condition, and crop management. So put together, this is actual environment, and then the second important thing to deal with noise. So probably our role as a phenotyper is to deal with this noise, and apart from dealing with the actual environment. So quantify and factor G by E in phenotyping. If we are able to do this effectively, our efforts are really successfully picked up by the crop improvement program. Professor Jose Luis already showed this slide where the relation between control over environment and relation with the target population environment is unfortunately opposite. So if we are hitting this way, very controlled environment, probably we have very poor relation with target population environment. If we go directly on farm trials, then probably we have no control whatsoever in terms of environment. So in CIMMYT, we settle down somewhere in between where we have fairly good control over environment, what we call managed environment trials, and we try to simulate the target potential environment. So all abiotic stresses, we always do in managed stress condition. It is not in growth chambers and very controlled condition, but it's fairly simulated environment. In terms of source of field variability or error, you can easily identify there are two types of errors: introduced error and inherent. So introduced error, of course, to be avoided, because there is no statistical tool which can deal with a very weird kind of experiment. But inherent error, we have to manage. So we can put together these two, and certainly this part has to be avoided. Otherwise, you have a statistical tool and models to deal with this kind of thing, but actually sometimes it takes away from the realities. So if this is the situation, like we use a different tool to look at the field variability before getting into the phenotyping business, and if this is the situation, certainly as our statistician Pancho Crosa used to say that you guys should consult me before planting the trial. Otherwise, you bring dead body and say that what you want eventually, because you have done already the experiment. So this kind of situation is there, so we can really organize the trial respecting spatial variability to avoid field gradient and minimize error, and use covariate analysis as needed. Sorry, what I did. And the second thing, a drought-tolerant maize is one of the flagship product of CIMMYT, and it's a history, a lot of people involved in this. The school led by Greg Edmis, of course, Scott Chapman was also part of the team. One issue, it's a simple example in case of drought trials, when to stop irrigation for getting a reasonable targeted level of drought stress. So most of the time we talk about 2 weeks before flowering. But we experienced several times that either drought was too much or too less. So we started looking at other options, and recently we started focusing on the GDD. If you look at, because this is in case of a particular phenotyping site, a particular maturity group, we identify that 550 GDD is the right time for applying drought stress. But if you look in terms of days, that 550 in one year, it was achieved in 48 days, and in another season, they say 550 was achieved in 56 years. So it's a 8 days difference. So if we follow this days, certainly that was the reason we were missing that. The second important cutoff was like when to resume irrigation. When we decide that, okay, enough drought. And I have seen many a time people take a lot of emotional decision on this. So we try to find some tool, and this soil moisture profile probe placed in the field. This is the field layout, and in each bed you will find one tube is placed respecting the field variability in terms of moisture and soil. And then we follow the soil moisture at the level of 40 to 60 centimeter deep in the soil, and look at when at that point, soil is hitting, moisture is hitting permanent wilting point, we say, okay, enough drought. So this strategy we are following for last 4 or 5 years, and it is really working very well. We can control our drought trial remotely rather than going at the site and even taking some decision which is ending in a wrong way. So with this small, simple tools, we are able to manage drought phenotyping in a way where we can identify the genotypic variability in terms of phenology and in terms of final harvest. Heat stress, we started this project recently, and certainly because in breeding program we have to deal with large number of genotypes, so we carefully identified the sites and then planting time. So these are the phenotyping network. This project is funded by USAID, what we call Heat Tolerant Maize for Asia project. And first year trial, we planted at all these locations, looking at the situation where we have more than 40-degree temperature for at least 2 weeks during flowering time. But what we found is that, okay, 40-degree temperature was there at all the locations, but the humidity was really playing a big role. So even if all these locations were having 40-degree, but mean yield of the trial was quite different. Then we looked at the VPD and clustered those locations in terms of VPD, which is including the temperature and humidity, and we were able to really pin down those kind of variation and identify the genotypic variability for heat stress. Secondary trait certainly is important, and we have to do this. But it is not just sufficient to know the secondary trait is related to stress tolerance. Probably it is important to know the breeding progress. As I said at the beginning, part of breeder wish list. It should have a proven breeding value. This some standard thumb rules given by Greg Adams is still very valid, and we really follow this strictly, that genetically correlated with yield relationship is causal, not casual. High heritability than gradient under stress, so that we are dealing with less G by E. Genetic variability for the trait must exist, otherwise there is no possibility of any selection. The trait should not be associated with poor grain yield. For example, if you select bird for early maturity, certainly it's a escape, but it's related to low yield. So this is, if the heritability of the trait or yield is high, there is no need of going into secondary trait. But if heritability is low for grain yield, especially in case of stress conditions, then certainly this selection on the basis of rather index selection has helped a lot in selecting the right genotype. Recently we started this root phenotyping. We are based in ICRISAT campus where they already had root phenotyping facility, so we developed a kind of replica of the same facility, little bit modification, and working on both functional and structural trait. Phenotype this association mapping panel, which clearly indicate a lot of variation in terms of structural trait like root depth and root dry weight, and more importantly this root functional trait, particularly water use and water use efficiency. This morning there was some concern about root growth pattern in this controlled condition. Of course it's a very directed growth in terms of growth pattern, but experience from ICRISAT, Vincent Vades and group has done is these functional traits are quite heritable, even if it is a controlled and directed growth. Of course the structural trait depends. Root goes where there is a nutrient and water. So we are focusing more on the functional trait, and using this strategy, we identified some drought donor lines. As Professor Roberto Tubosa this morning said that one size never fit to all. So these kind of genotypes, we know that they are not following the same mechanism, but they are all drought tolerant donors, and we have options to offer to the people in terms of drought tolerant donor, depending on the target environment, soil depth, and pattern of drought stress. So thank you very much for your interest. Thank you.
Thank you very much. Time for a question or two. Back corner.
Thank you very much for your talk. Enjoyed it a lot, not because you quoted me at the end, but because you kept it simple and down to the basics, because we often we get mesmerized by all the tools we have. It's either a question, but it's also implying a hidden comment on my side, but I would like to hear your comment, because rightly so, in the other talk, we heard a little bit the conundrum between association mapping versus the mass-assisted, marker-assisted selection with the major loci. So my question is, how many eggs are you putting in the basket of genomic selection at the moment at CIMMYT, particularly when it comes down to drought, because it's a perfect candidate because of the low heritability. But what is your view? I never like to put my eggs in the same basket, and I think there is a lot of reasons not to do that, like the previous speaker said. I would like to hear your comment. Thank you very much.
Yeah, that's a very, very good question, and I think that was a kind of discussion when Gary Etlin was leading our, breeding strategy, and he initiated this, genomic selection things, and we embarked upon several, projects on genomic selection. So, first of all, in case of genomic selection, the kind of population structure we are using, we don't go with biparental population, because particularly for a polygenic traits like drought or heat stress, really as these list of 10 genotype, they have a different mechanism. So we are going with 2 parental lines. It ends with a very narrow genetic base for that trait. So we are following multi-parent population. Like in case of Erie, they were using magic population. So it's a kind of 8 to 10 lines. So we don't say magic population, we say multi-parent synthetic population. And that population structure itself is so powerful, because at F2, F3, when we phenotype those progenies, we get a kind of very good options for taking forward. GWAS, basically we are using, between GWAS and GS, genomic selection, they are complementing each other. Particularly GWAS, we are using as a training set. So the genomic region, the favorable alleles and those things identified through GWAS, we use as a support for genomic selection, and that helps identifying the genotypic estimated breeding values, and those GBVs we use in case of genomic selection. So quite a few projects, but certainly we are keeping balance between GWAS and genomic selection. Yeah.
Okay, one last question from the middle of the room.
There are actually 2 questions, but
okay, I'll let you work with that.
If you can elaborate a little bit more on what you mean by precision in phenotyping, because I think that I understood something from your introduction, but then I got slightly lost when you elaborated a little bit more on precision. So I would appreciate if you can clarify again.
Okay. In terms of precision, as I said, there are 2 aspects of precision we are talking about. One, precision in experimentation itself. So as I said at the beginning, that we fixed a cutoff that if heritability of the trial is less than 0.4, we are not going to use that trial any kind of, whether it is GWAS or genomic selection or even conventional selection. And if it is optimal condition, 0.7. We struggle a lot, what is the value we should fix, but that feedback came from breeders, molecular breeders, that below that, it's like, it's a lot of compromise, and in these cases, if you do compromise, you compromise in future. So that is one precision. The second precision we are talking about here is a precision in, taking the observations, data recording, because for me, the important thing, first is the most important. If your trial heritability is not good, lot of variation, lot of introduced or inherent errors are there, whatever tool you use, you are observing what is already wrong there. So that is the key precision we are talking about here. Yeah.
Okay, thank you very much.